My name's Ian Copple. I'm Professor of Pharmacology and Toxicology here at the University of Liverpool. I run a research group called the Stress Response Lab, and we're interested in how cells of the body respond when they're exposed to things like medicines, chemicals, environmental agents. And we're particularly interested in how these stress responses can influence whether a medicine has a beneficial or a harmful effect on the body. And because the liver plays such an important role in how our body processes things that it might be exposed to, particularly medicines, a lot of our work focuses on how the different cells of the liver respond to different forms of stress and how we can use that to improve our treatment of disease. The Human Liver Research Facility here in Liverpool is built on over 10 years of collaboration between academics based at the university and also liver surgeons based at the Royal Liverpool University Hospital, which is just over the road from the lab. And we have access, through the generous consent of the patients, to excess healthy liver tissue that would normally be discarded at the end of surgeries. We're able to bring that back to the facility. We can either use the tissue or the cells within it for our own research, or we can make that available for other researchers to do the cutting edge experiments on human liver cells and tissue. Our bigger vision is to transition the entire facility away from the use of animal-derived materials in the medias and reagents that we use to keep the various liver cells and tissues alive in the lab. And if we can do that, that would be a real step change in what we do and potentially what others do working in this area, where we can continue to access and use the human liver cells and tissue for our research, but avoid the ethical and scientific concerns over the use of these animal-derived biomaterials. Our current project is all about trying to find alternatives to the use of FCS for growing human liver cells in the lab. Now, traditionally, these cells are grown in flat 2D cultures on plastic dishes, but we grow these cells in tiny 3D structures that we call spheroids. And these have been shown to better reflect how liver cells behave when they're in the body. And so we're exploring ways in which we could replace FCS in the media, but still maintain the liver-like behaviours of those cells outside of the body. These liver spheroids are used not only here, but by many labs throughout the world, both in universities and in the pharmaceutical industry. So if we can find reliable and robust ways to replace the FCS in the media that's used to culture these spheroids in the lab, there's opportunity that lots of groups and companies could transition to replace that in their own work, and that would bring real ethical and scientific benefits to research across the world.
Hi, I'm Rowena Sison-Young, and I'm the manager of the Human Liver Research Facility here at the University of Liverpool. The main motivation for doing what I do is being able to facilitate access to really precious human samples to researchers with an interest on human health. I think it's only right for these samples to be accessed by scientists who want to improve our health as a nation. And I think this is what motivates me the most. Animal-free research, I think, is important because it's almost giving animals in the lab a voice, a representation. It's almost making sure that the research that is done is relevant to humans. If you're using human-specific models in the lab, then the outcomes of your research is more specific, is more relevant than using an animal-based model. So I guess there are two benefits for the animal welfare, but also for the benefit of public health. I think the work that we do here at the Human Liver Research Facility is going to make an important impact on the way things are going right now with regards to research in human health. There are lots of different groups across the world, including us, who are developing these new approach methodologies, these new human-specific models. And to be able to do that, we need access to human material, and hopefully we can facilitate that. And therefore, we are somehow contributing to that movement into steering away from animal use and moving more into human-specific research.